Hello, everyone. Welcome to our talk. We're doing a tag team talk today, and I'll kick this off by giving an overview of our research. I won't be presenting new results today, but rather getting you thinking about different ways in which images can be used in drug discovery. This audience certainly needs no convincing that images are just an amazingly rich source of information. Images like these are really a work of art, but what's even more amazing is the amount of information that they contain, which is so much more than what meets the eye. But I'm always amazed by what often seems like an unreasonable effectiveness of images in representing biological state. How is it possible that pictures like this can encode so much information as to convey the mechanism of action of a drug, or the pathway of a gene, or intricacies of a disease state? We know, however, that at some level this is true. If you consider classic genetic screens, seeing the same phenotype suggests that genes in the same pathway were perturbed. So the idea is to take the basic concept and scale it up. And one dimension in which to scale it up is to paint cells with as many dyes as you can fit into a single assay. The talks today may have already mentioned the cell painting assay that labels many different cell constituents using cheap dyes that are easy to work with image-based profiling. Using assays like this, it's feasible to also scale up the number of perturbations. You can profile hundreds of thousands of samples, and this is a scale that is unmatched by any other profiling technology. Now, data of the scale gives you the means to address just so many problems across many domains, including drug discovery and development, functional genomics, and others. You've probably already heard many of us in this community talk about how we've applied this to problems like annotating the function of unknown genes and alleles or finding the mechanism of action of a compound. And all such applications are inherently about inspecting relationships among the perturbations and then hypothesizing something about a property of these perturbations based on these relationships. We typically rely on a guilt by association approach where, for example, if two compounds have similar profiles, then we expect that they're doing the same thing to cells and perhaps have the same mechanism of action. But today's talks focus on a slightly different set of problems. We aren't focusing on the relationships between the perturbations per se, but rather have the explicit goal of creating predictive models from these data. So Juan and Greg are going to talk about models we built using cell painting data to predict the performance of perturbations on other assays. Juan will talk about predicting the performance of a broad set of assays given cell painting data, as well as other data sources. And Greg will talk about predicting various cell health metrics given cell painting data. But I'll touch upon just a few other projects in progress that share the same general concept of creating predictive models, although with very different applications. In this project with Nico Taipale at University of Toronto and several of us at the Broad, we are aiming to create predictive models that may help us find cures for rare diseases. There are more than 7,000 diseases categorized as rare disorders, of which 4,500 are known monogenic. As many of you may already know, only 6% of these have FDA-approved treatments, and the path forward hasn't been clear. The traditional approach of using multiple assays and technologies to study each disease will be impractical. I should say that the path forward actually is clear to many of us in this community. Taking a phenotype first-based approach powered by image-based profiling, which allows you to scale across multiple diseases at one shot. The path forward is to create models for each disease that can predict whether a compound is reverting the phenotype. The Taipale lab has created an image-based profiling dataset for more than 1,000 Mendelian diseases across more than 3,500 mutant alleles by overexpressing them in a cell line. The initial goal was to systematically characterize the protein localization of disease variants by scoring images by eye, but we're now starting to look at not just protein localization, but the broad set of structural changes captured by cell painting. We can build predictive models based on mutant and wild type profiles to report whether a compound is reverting the disease phenotype. But what's really exciting is that we've set out to do this in a highly scalable way, teaming up with our colleagues at the Broad who've developed technologies that enable doing cell painting in a pooled format. That is, for each perturbed cell, we can read out both the cell painting profile, but also the barcode of the overexpression vector that entered the cell. This allows us to test out hundreds of alleles, so that's hundreds of diseases against a drug in a single well. In this project with Tarun Kapoor at Rockefeller, we are aiming to create predictive models of drug resistance and sensitivity. We take a set of paired cell lines where one of each pair has been modified to render resistant to render resistance to a specific drug, and we then profile the cell lines before and after drug treatment using cell painting. Then we investigate whether the drug-resistant or drug-sensitive cell lines have any morphological traits in common. That is, are there morphological markers of drug sensitivity or resistance? If we find such markers, this could be useful in predicting drug susceptibility for individual patients' tumor cells. And finally, in this project, we have collected cell painting and high-throughput gene expression data across thousands of compounds and several hundreds of genetic perturbations. And we're aiming to do two things. Going in one direction, we are creating predictive models of expression levels of individual genes given the cell painting readouts. In practice, this appears to work for only a small subset of genes, but importantly, these are now readouts that we're getting for free given imaging data. And going in the other direction, we're building deep learning models that can predict what a cell may look like if we modulate the levels of individual genes. Conceptually, this relates to our previous work where we've synthesized images of cells treated with increasing doses of a compound. This gives us a tool to understand how the expression of these genes relates to cell morphology. So that's an overview of our projects that broadly share this theme of taking a supervised learning approach with imaging data. And I'll now hand it over to Greg and Juan, who will talk more in depth about projects that they have led in this space.